Hello, grade 9 students. Here we start with Introduction to Genetics. We have to identify the carrier of genetic information. We have to study the laws of heredity and learn about the transmission of the traits. First of all, let's talk about the reproduction and genetics. First, what is reproduction? Reproduction is the biological process that leads to the increase in the number of individuals in every species. This is done to avoid the extinction of these species. So let's talk about some of the symbols. This symbol is called for the male, and this symbol is for the female. X meaning we have mating or we have crossing. The mating of the sperm cell and the egg cell, the male gamete, which is the sperm, and female gamete, which is the ovum, leads to formation of the zygote. This is called fertilization. So what is fertilization? Is the union of the male gamete and female gamete. This is the sperm and the ovum respectively. So as I said, fertilization is the union of one sperm cell with the ovum to produce a zygote. So one sperm plus one ovum gives a zygote. Sperm plus ovum leads to a zygote. So let's start with heredity. What is heredity? Is the transmission of traits from parents to their offspring. What is genetics? Is the science that studies heredity. So first of all, let's talk about the carrier of genetic information. All of you decided that the nucleus is the carrier of the genetic information. And to be more specific, the chromosomes inside the nucleus, they are the carrier of genetic information. Let's talk about genes. Genes, they are part of the chromosome. They are responsible for the appearance of specific traits, like the hair color gene, the skin color gene, the eye color gene. Every gene has a specific position. This position is called the locus. The two sister chromatids, they carry the same gene at the same loci. So alleles, they are versions or they are forms of the gene. Like her gene of the eye color, we have brown eye color, green eye allele, hazel allele, blue allele, and others. Gene of the hair color, we have brown allele, blonde allele, red allele, black allele, and so on. So this table is differentiating between the types of cells, mainly between somatic cells and the sex cells. Somatic cells, they are diploid. They are found on all body cells except the gametes, while the sex cells, they are haploid. They are found only in the gametes, sperm and ovum. Somatic cells, they are diploid, meaning that we have two copies of each chromosome, while haploid, they have one copy of each chromosome. So di meaning that we have two copies of each chromosome. Number of chromosomes is 2n. Number of chromosomes in the case of haploid it's 1n. In humans 2n equal 46. In humans 1n equal to 23. So this we have one copy of each chromosome. So here we have two copies. Sure, this one is haploid. While here we have two copies. Sure, it's diploid. So we have number of chromosome 1n equal to 4. Here it's 2n equal to 6. Alleles, dominant allele and recessive allele. Dominant allele is the allele that will be expressed in the case of homozygotes and heterozygotes. That's why it's called dominant and it's usually symbolized by a capital letter. While recessive allele is expressed only in the case of homozygosity and it's usually symbolized by a small letter. Genotype, definition, example, none, example. So, definition of genotype is the set of genes and their corresponding alleles, while phenotype is the set of observed traits that we observe that are resulting from the expression of the genotype, like tall, short, black, and so on. These are the phenotype. And here we move to the type of crosses. One of these is the test cross. Test cross is done to identify whether the parent is pure or it's hybrid, meaning that if it's homozygous or heterozygous. It's done between a dominant individual with a recessive individual. So if the dominant parent is pure, so it should give one phenotype in the offspring. While if it's hybrid, it will give two phenotypes in the offspring. Type of crosses we have: number one, it's the self cross. Number two, we have the monohybrid cross, and number three, we have test cross. Self-cross between F1 crossed with F1. This is called self-cross. Monohybrid is done between two individuals that differ only when traits and they are pure. Tessicross is done between a dominant and a recessive individuals. Case of heredity, we have dominance. One dominate the other. Co-dominance, both are expressed. And intermediate, which is the color of intermediate, is called incomplete dominance. Activity 3, carrier of genetic information. These karyotype. Karyotype is the arranged set of chromosomes. In diploid homologous chromosomes, they are arranged according to the following criteria: decreasing order of size, position of the centromere, and the banding pattern. This is the karyotype shown in this figure. Gonosomes. What are the gonosomes? Gonosomes, they are the sex chromosomes that determine the sex of the individual, X and Y chromosome. If we have two X, so it's a female. Y, if you have X and Y, so it's a male. Autosomes, they are all the chromosomes except the sex chromosome. They are 22 pairs of autosome numbered from 1 to 22.